talk here for Sue. And again, I appreciate everybody sticking around. It's been a great session so far. Okay, you got your talk up?
So far, yeah.
So far.
Pressure's on. Okay.
Hopefully I won't start coughing too much.
You want to save it?
No.
Okay. Oops. How did I get in the front here?
Okay.
Yeah. Okay, well, I thank everybody for sticking around, and I've enjoyed all the presentations. What I'm going to talk today about is a little sort of a combination of fluorescence and electrochem. And a lot of this work was actually inspired by Christian Amatori's work looking at peroxynitrite and nitric oxide using the in vivo amperometry. And what we're trying to do is do a similar approach, but using a separations instead of triple step amperometry to detect the reactive nitrogen and oxygen species. So we got interested in this primarily since some of our collaborators at the University of Kansas are working at on the blood-brain barrier and the Neurovascular Unit. And we're really sort of interested in looking at the induction of nitric oxide synthase at the blood-brain barrier, which causes the opening of the blood-brain barrier, the nitric oxide release. And we're also interested in macrophages and microglia, which are the immune cells in the brain and the blood, respectively, that will also produce not only nitric oxide but reactive nitrogen species. So just sort of summarize on what's known about this. The NO does increase the permeability of the blood-brain barrier. I'll show a slide. The next slide will show the actual changes in permeability for sucrose when you add NO donors. And then it was also found by Autus's group at KU that if you add cytokines to blood-brain barrier microvessel endothelial cells, which are a mimic of the blood-brain barrier, it will induce nitric oxide synthase and cause release of NO by these cells. And another thing that's interesting is that if this happens and there's also superoxide being produced, then you'll form peroxynitrite, which can then nitrate proteins, cause DNA damage, and then lead to cell death. This just shows sort of the rationale about this nitric oxide. So you can see here, if you add these NO donors to the blood-brain barrier microvessel endothelial cells, you can see the permeability significantly increases for this is either mannitol or sucrose. So nitric oxide is important for looking at transport of compounds across the blood-brain barrier. And of course, if there's ischemia or stroke, you're going to release nitric oxide and other compounds, and so you're going to see an increase in blood-brain permeability. So the goal of our research right now is we're doing this collaboration with Chris Culbertson at K-State, who's an expert in single-cell analysis, and was one of the first people to do single-cell analysis in Mike Ramsey's group, I don't know, 10 or 15 years ago. And the idea is to take our electrochemical methods in conjunction with his single-cell analysis system and to actually see if we can analyze these reactive nitrogen and oxygen species in single cells. And the cell types we're looking at are macrophages. We're looking at Jurkat cells, which are lymphocytes because they're non-adherent cells and they're easier to manipulate in the single-cell analysis system. And then eventually we'd like to look at microglia. And some of the analytes we're looking at are nitric oxide, peroxynitrite, nitrite, and then hydrogen peroxide. So we're using microchip electrophoresis for these experiments, and there are several reasons for this. One is that the small injection volumes are compatible with single-cell analysis. The real key reason is that we can use a separations-based method to actually separate out the different reactive nitrogen and oxygen species from both interferences in the cell and from each other. So we can do multi-analyte detection. And because this is in a microchip format, we can do this very, very quickly. So we can detect and separate things in a 30-second time scale. So if even short-lived species we can detect using the microchip approach. So I'm going to show some examples with both fluorescence and electrochemistry. We've used fluorescence to sort of check the electrochemical detection methods. And then you can do high-throughput analysis in this single-cell analysis system. Again, we're using electrochemical detection is our goal for this project. And there's several advantages for this. We can integrate the electrodes directly into the chip. It's selective, so we're only detecting redox active components in the cells. And if you're looking at reactive oxygen and nitrogen species, those are the important components that you want to be able to look at anyways. We can put multiple electrodes and do voltammetric characterization to further identify the metabolites. And then, so this is basically what I already said. So, okay, so in this talk, in the electrophoregrams I'm going to talk about today, we're using primarily in-channel detection. So we're placing the electrode at the end of a separation channel in the microchip format. There's also N-channel detection. I think Adam Woolley's here, and he was the first person to do this with Rich Matthews way back in 1980-something, I think. And then, but right now we're using a working electrode right at the end of the separation channel. And what we use is a isolated potentiostat that we have from Pinnacle Technologies, so we can apply the separation voltage across the channel and still detect things without wrecking the potentiostat. And the advantage of this approach is that you can actually get better separation efficiencies because you have the electrode in the channel. So this just shows some of the species that we might find in our cell lysates: nitrite, tyrosine, ascorbic acid, and glutathione, and hydrogen peroxide, separated in channel and end channel. And you can see the resolution is not very good if you place the electrode outside the channel. But if you use this in-channel separation method, you get a much better separation efficiency. One of the, not drawbacks, but one of the considerations for in-channel detection is that you do have to readjust your electrode potential for detection to take into account the separation voltage, which will influence your actual detection potential. And again, you can get very nice, you get better separation efficiencies using the in-channel versus end-channel detection. So I won't. This whole session has been about nitric oxide. So as everybody suggested, it's a very important molecule in neurotransmission, immune signaling, smooth muscle relaxation, and we're really interested in more the destructive effects, how nitric oxide reacting with superoxide will form peroxynitrite or other reactive oxygen and nitrogen species and cause programmed cell death. There's a lot of different species. The main ones will be, we're concentrating on our peroxynitrite, nitrite, nitrite, and then NO that's in here somewhere, I think. Yeah, right here. So the first thing that we wanted to do is just see if we can separate all these different compounds and detect them electrochemically with microchip electrophoresis with electrochemical detection. So all these separations are done in reversed polarity using TTAB as a surfactant and in the microchip format with in-channel detection. And this just shows we wanted to find out where the nitric oxide came out and the nitrite come out. And so Dulan Gunasekera that's here and Vakasi da Silva, who's also over here, worked together on trying to identify where these peaks came out in the microchip format. So they used proline NONOate, and this just shows the reaction over time of proline NONOate, where it's producing nitric oxide, and you get nitrite as well. And then you can see the reduction of the proline NONOate, which is electrochemically active, and then the increase in the amount of nitric oxide. You can actually also see the increase. Well, you see an increase in NO, and then it starts to degrade and actually form nitrite. So you can also follow that using the microchip. The other compound that we're really interested in, and the reason we really got excited about the work by Christian Amatori, is we were really interested in looking at peroxynitrite. Christian Schoeneich at KU has been looking at peroxynitrite a lot to look at nitrotyrosines on proteins and oxidative modifications of proteins. And there was some interest to see whether peroxynitrite was being generated at the blood-brain barrier and actually nitrating proteins in the endothelial cells and causing cell death. So as people have mentioned, peroxynitrite has a very short half-life at physiological conditions. There really are no very good fluorescent probes for peroxynitrite. Most of them cross-react with other compounds. And so the only way you can detect it is either with UV detection. It does absorb at about 400 nanometers, or do electrochemical detection, as Christian showed in the first talk. Well, one of the things that a student in my group discovered Celeste Frankenfeld quite a while ago is that if we take peroxynitrite and keep it at a high pH, then we can actually separate it and detect it using capillary electrophoresis. So when you buy peroxynitrite from Cayman Chemicals, it actually comes in a solution at pH 11 or 12, and so it's stable, obviously, during shipping. So what she was able to do is separate, this is a Cayman Chemical peroxynitrite. She was able to separate peroxynitrite from nitrate and nitrite, using CE with UV detection. So this got us excited. We thought, well, maybe we can take this peroxynitrite and keep it stable in a microchip at a high pH and detect it electrochemically in a cell, in a single cell. So this shows some work by Matt Hulvey and Dulon, where they looked at the peroxynitrite degradation using microchip electrophoresis with electrochemical detection. So again, this is reverse polarity. So you can see the peroxynitrite is decreasing over time. And then you have also a peak for nitrite in the electrophoregram. We can also generate peroxynitrite using SIN1, which is a peroxynitrite generator. And so here it forms nitric oxide and superoxide and generates peroxynitrite. And you can monitor the generation of peroxynitrite over time. These are subsequent injections using microchip electrophoresis with electrochemical detection. And so we were able to monitor the amount that was produced in 1 minute intervals over approximately an hour here. So this got us believing that we can really start detecting this in single cells and bulk cell lysates. And so one of the things that we were interested in looking at is macrophages, because that's sort of a model system for reactive nitrogen and reactive oxygen species. And so the idea is to take the macrophages, get them to produce nitric oxide, see if we can detect the nitric oxide using this microchip approach, and then add a stimulating agent to produce the superoxide to form the peroxynitrite, and then also see if we can detect it using the separations-based approach. So we've looked at two different types of cells. Jurkat cells, which are not inherent lymphocytes, which Chris Culbertson has quite a bit of experience with in the single cell analysis system, and then also these raw 264.7 murine macrophages. And then these are some of the different reagents that we use to induce both inducible nitric oxide synthase and NADPH oxidase. So first experiment that we did was with the macrophages, the raw 264.7 macrophages. And as I said, these can be activated to produce both nitric oxide and peroxynitrite, but they are adherent, so there can be some problems with stickiness, although we found that you can actually buy some cell lines that aren't quite so adherent that will be easier to use in the single cell analysis system. So the first thing we wanted to do is just see, can we lyse these cells and detect nitric oxide, but also look at what else is in the macrophage cell intracellular constituents in the macrophage. So the approach was that they took macrophages, they stimulated them with LPS, which we've talked about quite a bit in this session, to induce nitric oxide synthase, and then you incubate for 6 to 8 hours, harvest the cells, then we centrifuge the cells, and we lyse them in this high pH buffer that we're using for the separation, and then centrifuge the lysate with a 3 kilodalton molecular weight filter, and then we inject it into the microchip. So first of all, you can see this is just a native cell lysate. So we do see, we get a peak for nitrite, tyrosine, glutathione, and I'll show in a minute a peak that co-migrates with NO. Whoops. So if you look at the actual electrophoregram, what you see is because because this is a cell lysate and we're just putting it in the sample vial, what we see is the NO decreases over time because it's basically diffusing out of the cell. And then you can also see that there's an increase in the amount of nitric oxide that we see in these stimulated macrophages versus the native macrophages. We then have repeated this experiment with Jurkat cells. These, again, are non-adherent cells, but they can also be activated by LPS to produce nitric oxide. And as I said, they're a good model system for the single-cell analysis system. And here, again, this is just the cell lysate, so you get some peaks for cysteine, tyrosine, and glutathione, nitrite, and the nitric oxide. Again, you see the native cells are not producing very much nitric oxide at all. And then as you put the lysate into the well, you can see the nitric oxide dissipating out of the well, but we can definitely detect the presence of nitric oxide production in these cells. So in order to verify that we were getting nitric oxide production and determine approximately how much nitric oxide we were getting produced in the cells, we used the fluorescent method using DAF-FM diacetate. The DAF-FM diacetate is, as Dana indicated, or actually the DAF-FM reacts with the nitric oxide to form this fluorescent derivative. And we also put in this cell another fluorescent probe, CFDA, as sort of an internal standard. So what we wanted to be able to do is normalize our fluorescence for our nitric oxide to something that was inside the cell that would not be affected by any reactive nitrogen or oxygen species. So this would normalize cell volume as well as the number of cells in the bulk cell lysate. One of the concerns was that we would also detect the product of the reaction of DAF-FM with dihydroascorbic acid. Sweetler's group has several papers on this where you can get false increases in the amount of nitric oxide that you think you have in a cell due to the reaction of dihydroascorbic acid with the dye. So one of the advantages of using microchip electrophoresis is that you can actually separate out this product with dihydroascorbic acid from the product with the nitric oxide and the internal standard that we're putting in the cell. So we did the same experiments that I just showed with the this is actually with the Jurkat cells, but we did the same experiment that I just showed with the electrochemical detection, except in this case we loaded the cells with the DAF-FM-DA and the CFDA, incubated them and then lysed them and then centrifuged them and put them in the microchip with the fluorescence detection. And here you can see that you get here's the native cells and the stimulated cells. So here's the production of nitric oxide. At first glance, it looks like the native cells, well, if you look at the ratio, the stimulated cells are producing more nitric oxide. But another thing that we found is that since nitric oxide is toxic, about half these cells actually died. So when we were doing this study, Emily Mines, who did a lot of the fluorescence work, who's now at University of North Carolina, she counted the viable cells and then for each of these before we quantitated how much nitric oxide was actually present intracellular into the cells. So here's the, let's see, does it have a concentration here? Whoops. Sorry. Oops. So the concentrations here was about, the ratio was 0.69 to plus or minus 0.02, and there was about a 2.3-fold increase in the amount of nitric oxide that was produced. Now we could actually calculate how much nitric oxide was produced. We used one of the NONOate salts to make a calibration curve of the NO concentration versus use electrophoregram to determine the amount of calibration curve for NO, and then we could quantitate the amount of NO production in the Jurkat cells. Now this is integrated over the period of the incubation, so we got about 600 nanomolar for the native cells and about 1.5 millimolar for the stimulated cells for the NO production, or micromolar, sorry, 600 micromolar. So again, we see about a 2.5-fold increase in the amount of nitric oxide that's produced. The other thing we wanted to look at is the peroxynitrite production, and this is very preliminary data. But we took the macrophage cells, and we did the same protocol that we used for production of nitric oxide, and then we added PMA. We added PMA to induce the nitric oxide or the NADPH oxidase. And here what we should get is the production of peroxynitrite. And so actually what you see here is when we take the lysate, we see nitrite. We see a peak that we believe is that it co-migrates with NO. Whoops. And then we also see a peak here. Now this is at a high pH, so we lyse these cells at a really high pH, at like pH 11 buffer. And we have to also see that the T-tab seems to interact with the peroxynitrite and stabilize the nitrate, maybe from the hydrophobic interactions. But what we can actually detect a peak in the lysate that co-migrates with peroxynitrite, and it also has similar voltammetric properties to peroxynitrite. So we're hopeful that we'll be able to do this in the single cell analysis protocol. So in addition to doing the detection of peroxynitrite and nitric oxide and nitrite with the electrochemical detection, we've been working with Chris Culbertson to look at the amount of peroxynitrite or the amount of nitric oxide that's produced in single cells. So here he has a microchip where you bring the cells into the channel, and then basically the cells are lysed by the separation field at the beginning of the channel, and then the contents are separated. So basically this shows just a schematic or sort of a sequence of the single cell analysis. So first the cells come into the chip, then they reach the separate or the lysis channel. They're lysed, and the contents are then injected into the separation channel where they can be separated, and then the cell debris will move off into a separate channel. So this shows a couple of electropherograms for native versus stimulated cells. So you can see here, so when we were doing the bulk cell lysis, we actually converted all of the CFDA, due to the base hydrolysis, all the acetate groups were gone. But here you actually have a monoacetate that's still in the cell because we haven't, we lyse it here in physiological pH buffer. So here we actually have two basically internal standards. And I don't think I included the data in the talk, but Chris has shown that both of these standards, the ratio of these two standards stays the same, and the ratio of the DAF-FM to either of these is indicative of how much nitric oxide has been produced in the cell. So here we have the native cells, and he can do about 100 of these at, I think, 30-second intervals, or maybe even less than that. And then here's the stimulated cells. This shows the histograms for the NO production using the stimulated and versus the native cells. So I think I may have forgotten to. This is with fluorescence detection using the DAF-FM dye, which has been incorporated into the cells. And so you can see that you do see an increase in the amount of nitric oxide in the stimulated cells. He's also done some preliminary work with macrophages and is able to show there's an increase in the nitric oxide in those as well. This, again, as I mentioned, the ratio of these two stays pretty much the same. But if you take the ratio of the DAF-FM to CFDA, you can see that there's an increase in the amount of nitric oxide that's produced. And you can pick either peak, the CFDA2 or the CFDA1. And you can see the amount of increase that we're seeing due to stimulation is very similar to the amount of increase that we saw when we did the bulk cell analysis. So with the bulk cell analysis, when she was reporting the concentration, intracellular concentration, she basically calculated the amount of NO that was produced by all the cells, divided that, and then counted the number of viable cells, and then determined the approximate intracellular concentration for each individual cell. So these ratios come out very similar as well for the single cell analysis system. So I really whipped through this, but it's getting sort of late in the day anyways. But our ultimate goal here is to actually see if we can take microglia and put them, analyze them with the single cell analysis system. So one of the things we're doing right now with Chris is we're incorporating electrochemical detection into the single cell analysis system using platinum electrode and a PDMS substrate. And the idea would be to bring the cells into the single cell analysis system, lyse them in the channel. The amount of time, so they'd be lysed in a high pH buffer, and they'd only spend less than a second to be lysed, and then injected into the separation channel, where they would be separated immediately at a high pH. So we're hoping that because the amount of time between the lysis and the actual detection, and the fact that we're using a high pH, we should be able to capture peroxynitrite and some of these other reactive oxygen species in single cells using this approach. So the future directions is to integrate the electrochemical detector into the single cell analysis system, and Dulan's working on that, building a PDMS glass hybrid chip to do that. And then we want to compare the NO production in bulk cell lysates with single cells using NO. We can determine the NO production using fluorescence detection in the bulk cell lysates, but with the single cells, if we can compare what we're getting with fluorescence with what we're getting with EChem, we can then calibrate that. Also, we want to detect peroxynitrite in bulk cells using the microchip electrophoresis with electrochemical detection, and also compare that with a fluorescent approach. We have a dye that we received from a group in Hong Kong that is supposed to be selective for peroxynitrite. So what we'd like to do is the same comparison experiments that we did with nitric oxide with peroxynitrite. And then eventually measure intracellular concentrations of NO and peroxynitrite in single macrophages and microglia by looking at the response to specific stimuli. Now, one of the things is, one of the questions we frequently get asked is, what about the stimuli before you analyze the cells? But most of these cells are incubated, actually, in this LPS, and it takes 6 to 7 hours for them to actually start producing large amounts of nitric oxide or peroxynitrite. So it's our feeling that if you bring the cell into the channel after it's been stimulated for 6 hours in LPS or PMA, then the amount of change that you would perturb in the nitric oxide production or peroxynitrite production in that couple of minutes or maybe a minute that it takes to get into the device would not make a significant difference in the total amount of nitric oxide, intracellular nitric oxide or peroxynitrite that you would see. And then lastly, we'd like to, since we're looking at single cells, we'd like to correlate phenotype of microglia or macrophages. Christian showed, the activated versus non-activated macrophages. Also, microglia are activated by a number of different other substances. If there's brain trauma and some albumin gets into the brain or beta amyloid gets into the brain, that's known to activate microglia. So we would like to look at how those affect the production of reactive oxygen and nitrogen species in these cell types. So with that, I'd just like to thank the people that worked on this project. Matt Hulvey and actually Celeste Frankenfeld really got us started on the peroxynitrite. Emily Mines and Giuseppe did most of the fluorescence studies. Derek Jensen, along with Derek Jensen. Fracassi, Dulan's not on here because this is his slide. Dulan actually is sitting in the third row there, actually did most of this work. He's right here. Did all the EChem work here. And then also Pinnacle Technologies supplied the potentiostat. In Chris's group, Chris has been fantastic in helping us getting the single cell analysis stuff working. And his student Eva Meadow has done most of the single cell analysis, and Ann has been growing the cells at K-State. And then also Christian Schoenig that's helped us understand better the nitric oxide peroxynitrite chemistry. And then this is a research group here. And just to mention, Tom actually has a poster, or no, I'm sorry, a talk, 4:45 tomorrow. If anybody's looking for something to do really late on a Friday afternoon, he's given a talk on, or a poster on methyl arginines and nitric oxide. And then Jessica's given a talk tomorrow on some point-of-care testing for developing, or pharmaceutical testing for developing countries. And then Rachel has a poster tomorrow on some catecholamine analysis that you might want to take a look at.
Right.
So with that, I'd be happy to answer any questions. I tried to go fast so you could all make it to the bar.
Questions for Sue? I'd like to start with one if I could.
Yeah.
I had the question in my head, and then I definitely wanted to ask it toward your last slide.
Yeah.
And so you might be able to explain why or why not
Yeah.
do this. But like, so if we have the probes for the nitric oxide, and now you might have a probe for peroxynitrite. Again, it probably has to be characterized.
Yeah, fluorescent probe.
Yes, a fluorescent probe.
Yeah.
I'm sorry. If you have those two probes, and assuming that their emission wavelengths are far enough apart, now why not just flow cytometry versus the electrophoresis?
Well, one of the things that we'd ultimately like to do is combine our separation method with the things that Christian's doing with the chronoamperometry. So if we could, one of the things we'd really like to do is trap a cell, measure the amount of nitric oxide and peroxynitrite that's being produced extracellularly, and then at a certain point move that cell into the, or lyse the cell actually in the device and measure the amount of reactive oxygen and nitrogen species. So that way we could measure instantaneously the intracellular and extracellular concentration of peroxynitrite. One of the things that I always wonder about with fluorescent probes, at least in our hands, is there's always an incubation time, right? I mean, so you have to allow the probes to go into the cell first. So that's a kinetic step. Then they have to sit there for a while. And then you're really getting an integrated amount of peroxynitrite produced over time. I mean, you're really getting 10 minutes worth of peroxynitrite. So you don't really know what the flux is or the production is at any given time point. So with this, with the combination of amperometry and the separations based, you could look at the flux of the peroxynitrite or nitric oxide out of the cell, and then at any given point, lyse that cell and measure the intracellular contents.
Right.
And with the fluorescent probes, I think they'd just be too slow to do that.
Yeah, but you're right. Yeah, I think your first point is the one that I would definitely consider, is that the flow cytometry, at least when we do it, it's really good for looking at intracellular levels of some molecule of interest or analyte of interest. But if you want to actually see dynamic release,
well, the other thing is we can also look
more
at.
difficult.
It's true you could look at several different probes, but we're doing direct detection, and a lot of these probes, for example, the DAF-FM reacts with ascorbate. So if you're looking at microglia or some other system that there's a lot of ascorbate around, you could give really bad, inaccurate results for the amount of NO that you think the cell's producing.
Correct. Other questions for Sue? Scott?
Yeah, Scott.
When you guys transition to doing the lysis on chip with culverts, do you foresee any problems with electrochemistry? Does he have to pulse to pop the cells, like an AC pulse?
Oh, okay. I thought I had it in here, but we have a different schematic so that we don't have the high voltage at the detection end. Is that what you're getting at?
Yeah.
Yeah. No, I meant to put it in here, and I didn't realize it was. But yeah, we've reversed it so we don't have to, so we can ground the detection end, so we don't have to worry about that. But it's a good question.
Okay, if there are no other questions, please join me in thanking Sue, all of our speakers, especially Dr. Amatore, for flying overseas to present today. Thank you.